What do producers of lab-grown meat use as a medium to nourish the growing tissue? As far as I can tell, as recently as 2018 it was impossible to nourish the cell cultures in laboratory meat production without growth fluid containing animal blood. Articles today often note that producers have either been able to eliminate this practice or are moving away from it but are vague about exactly how, and about what they've used in place of those ingredients. So, what's in it? Does the process of growing meat really work without animal products other than the stem cells needed to establish the culture? I design cell culture media, which is what is used to grow lab meat as well. In our industry, we've moved away from using serum ORPSA in all of our new formulations. It's a combination of vitamins, amino acids, sugars, trace elements, buffers, and, in some cases, proteins. It depends on the meat origination source. A friend of mine was working with pig stem cells to grow meat. He had a very specific custom-made recipe for medium which worked. However, it also worked exceedingly well for a fungus as well. He had 3 years of all contamination of this fungus no matter what he did. I work at a biotech company in an R&D group centered around selling cell culture media to startups companies in this industry. The animal component most cells need is fetal bovine serum or FBS. FBS contains growth factors, lipids, trace elements, and some vitamins. Serum-free cell culture involves adapting these cells to lower concentrations of serum while replacing required components, e.a. growth factors from animal-free sources. Most of these growth factors can be produced recombinantly. From what I'm seeing, current state of the art is using bovine growth serum or, as you state, basically blood or blood-derived liquids, but a number of labs have indicate they're working on a plant-based growth medium. Cultured cells still need to be grown in some kind of medium like Dulbecco's modified Eagle medium with 1020 fetal bovine serum. Yes, there are synthetic alternatives to serum, but they are stupidly expensive and not as good. There are alternatives to FCS, but they are also very expensive. We grow cells in defined growth medium that has all the base ingredients but uses insulin, selenium, and transferrin. Many mammalian cells grow well in this FCS substitute after an adaptation period. If they can grow it and it include the exact same nutritional value as actual source meat, then I'm willing to give it a go, assuming they don't lace it with other stuff for our protection and whatnot. I don't want any experimental stuff added to my already experimental meat stuffs.